DNA replication. This video will cover how DNA replicates itself and makes an exact copy in your cells. Let's begin with a quick review of the structure of DNA. So you should remember that DNA has a double helix shape, which is that twisted ladder shape. It is comprised of two long nucleotide chains. The outside is a ribose phosphate backbone, that sugar phosphate group backbone that forms the outside of the ladder. And you also should remember that DNA is anti-parallel, meaning one strand runs one way and the other strand runs the other way, like a highway. DNA replication is also known as DNA synthesis, since new DNA is being made or synthesized. It's obviously clear that it occurs in the nucleus, since that's where the DNA is located. We also already discussed how DNA synthesis or replication occurs during the S phase of the cell cycle, so before mitosis. You should know why it occurs before mitosis. It occurs in three phases that we'll go over. And one of the key words for this section is known as semi-conservative replication, which means that when you make a new DNA molecule, it consists of one old template strand and one new strand. So every new DNA molecule that is made is half old DNA, half new DNA. Let us now go over the actual steps of DNA replication. You are responsible for knowing the names of the enzymes involved in DNA replication, if I say them, and you also are responsible for knowing the actual steps in order to replicate or synthesize a new DNA molecule. So step one is enzymes need to bind to the DNA strand that is being replicated. Step two, a specific one known as a helicase, unwinds the DNA double helix. You can see that at the bottom where the two DNA strands are ripped apart and they're separated, so it gives some space for the new strand to be synthesized. Step three is the new nucleotides are added by an enzyme known as DNA polymerase, since it is polymerizing or making new DNA. And it follows the base pairing rules. So the new strand complements the original strand. And you can see that at the bottom, you see that you have ACGAT, and then the orange nucleotides will fill in with the complement of TGCTA. This is how we're able to get an exact copy of the DNA. So you notice at the bottom right, you have two new strands of DNA, each semi-conservatively replicated, one orange strand, one green strand, and you notice the base pairings are exactly the same. So the DNA molecules are exactly the same to each other. Let's now discuss the geography of where the DNA replication actually begins. So you can see in the dead center is something known as an origin of replication, so that is where DNA is actually started. The entire little circular shape is known as a replication bubble, because it looks like a little bubble in the DNA. You notice that the original strand is the blue strands, while the newly synthesized DNA are the red strands. And you actually can notice that there's two things known as replication forks, one on the very left-hand side and one on the very right-hand side. So you can see, let me get my pen. So here is one replication fork, and here's the other replication fork. And this is where the DNA helicase is actually located. It is unwinding these DNA strands and separating them. If you notice, there's actually two different strands of DNA that are being made. One of the strands is being made continuously. You notice at the top on the left-hand side right here that the red new DNA strand is one continuous piece. However, if you notice on the other side, there's all these little chunks known as Okazaki fragments. DNA can only be synthesized in the 5' to 3' direction. Therefore, nucleotides can only be added to the 3' end of a DNA strand. So if you notice, if the replication fork is going to the left, you have the 3' direction on here, so DNA is being made from the 5' to 3' direction. However, you can't just do one strand and then do the other strand later. You have to do them at almost the same time. The other strand can only be done also in the 5' to 3' direction. Since it is anti-parallel, the other strand has to be made in the opposite direction. Well, notice if the replication fork is located right here on the left, there's no way to continuously make that molecule. You are forced to make small little chunks of DNA, and then later we're going to stick them together. So those are the Okazaki fragments. There's two different strands of newly synthesized DNA. You have the, excuse me, can't work my pen. You have the leading strand, which is the strand that is continuously made. So that is the one that's made in the 5' to 3' direction in one continuous piece. And then you have the lagging strand, which is the strand that is made in the opposite direction, still 5' to 3', however, and it is made of Okazaki fragments that later. So you can see on this DNA replication diagram, you're only looking at half the replication bubble. So you see that the overall direction of replication is to the left. You see that there's the replication fork where the helicase is unwinding the DNA and opening up the strands so the polymerase can add new nucleotides in the 5' to 3' direction. On this diagram, you should be able to identify the leading strand and the lagging strand. So as we just said, the leading strand is always continuously made. So you can see that the leading strand, therefore, is the one up the top here, because it's continuously made in the 5' to 3' direction. Therefore, the other one must be the lagging strand, which is still made in the 5' to 3' direction, but due to the space, it has to be made in little Okazaki fragments that then have to be stuck together to make a continuous strand of DNA. This animation shows how the Okazaki fragments of the lagging strand are actually linked together. So you can see the two little pieces are connected together, and then an enzyme known as DNA ligase forms the covalent bonds and links the Okazaki fragments together. So basically, you just have to stick the two ends together, the two sticky ends together, like two pieces of Velcro. They stick together, and then DNA ligase connects the strands together to make one continuous piece of DNA. Let's review the enzymes that we've discussed so far that you need to know. So first off, we have DNA helicase, which unwinds the helix like the replication fork. We have DNA polymerase, which binds the new nucleotides to the template strand and adds the nucleotides to the 3' end to make that 5' to 3' DNA synthesis. And then we just talked about DNA ligase, which then bonds the Okazaki fragments of the lagging strand together. So you are responsible for knowing the function of these three different enzymes during DNA replication.